Hello, my name is Leung. On behalf of my co-chairs, Giovanni Cacciamani and Carmen Jeronimo, we welcome you to this webinar on artificial intelligence, the role in research and patient management. We're delighted to have two excellent speakers with us today to present to us. Joining us with almost 400 delegates at this webinar. So before we start the talks, I want to just encourage our delegates to put in questions during the talks, and we can address them at the end of each talks. And also, we will have a short final discussion session at the very end. So I pass the floor to Gio to introduce our first speaker.
Thank you so much, Professor Leung, and thank you so much for the European Association of Urology for organizing this very interesting meeting. So the first speaker of today is Professor Van Halen. Dr. Van Halen is an associate professor of medicine at the Harvard Medical School and chief of the Division of Population Sciences at the Dana-Farber Cancer Institute and an associate member of the Board Institute of the MIT and Harvard. His research focuses on computational cancer genomics, the application of new molecular profiling technologies to advance precision cancer medicine, and studying resistance to cancer therapeutics. As both a computational biologist and medical oncologist, he has specific expertise in clinical computational oncology and in the development of algorithms to analyze and in making more clear the interpretation of genomic data for clinical focused questions. So today, Dr. Van Halen will educate us regarding the neuronal network for risk stratification in prostate cancer. The stage is all yours.
Great, thank you so much for the kind introduction. And before I begin, am I on the right version? Does it look good on the PowerPoint?
It looks excellent. Thank you.
Fantastic. So thank you for this introduction and thank you for the opportunity to present to this extraordinary group. Again, as you heard, my name is Ellie Van Allen. I'm a medical oncologist at Dana-Farber and I'm a computational biologist and with an interest in genitourinary oncology. Generally speaking, I'm going to talk to you about some of our work with biologically informed neural networks in prostate cancer. You can also follow along offline shamelessly. Here is my Twitter account where I make jokes about our hospital's electronic health record. So first, here are my disclosures. So over the next about 20 to 25 minutes or so, I thought I'd provide a little bit of background and motivation for that prompted the project that we decided to pursue, describe some of this sort of biologically informed networks that we've developed, and then demonstrate how we applied them to some of the same cohorts that we've been studying for years now to make some inference in prostate cancer, and then end with a little bit about how we like to take the energy from those experiences and use them moving forward to inform our research. But first, a little bit of background, sort of why we decided to do this. So about 4 to 5 years or so ago, as part of our Stand Up to Cancer Prostate Cancer Foundation Dream Team initiative, we organized about 1,000 prostate cancer tumor normal exomes and performed a genome-wide mutational significance analysis looking for genes that were recurrently mutated in prostate cancer. Irrespective of the clinical context, we wanted to see how many genes really were mutated above a certain background rate and a certain model rate that might actually be informative for biology. And what we found was sort of described in this figure here as part of a study that's cited down below, whereby we identified what we called a long tail of genes that were significantly mutated in prostate cancer. Some of these genes on this list might look very familiar to you. Some of them may not look familiar at all to you or to us. And a lot of them we know not very much about, and we'll have a lot of work to do as a community to try to make sense of how each of these things individually relate, to the oncogenic potential of prostate cancer. But one thing we actually wanted to do in this study that we kind of frankly, admittedly didn't really solve was to take these genes and not only just consider them in isolation, but rather try to map them to whether they were enriched in primary or metastatic prostate cancer. And specifically, which we summarized here, whereby we basically looked at the frequency of mutations in metastatic disease relative to primary disease in this cohort of 1,000 patients. And we, in essence, tried to map which ones were enriched in metastatic prostate cancer compared to primary. And the first thing we observed was actually something not very surprising. There were 3 genes very screamingly obviously enriched in metastatic prostate cancer, AR, P53 and P10. This is not new. This is sort of there was actually some jokes on social media about P53 and cancer genomics for those who follow that kind of thing. So that wasn't new, but that was reassuring that we could recover those things. But the hard part was that there was this blob of genes down below that we actually couldn't really make much sense of. Which of these mattered? If they mattered, did they matter in isolation, or did they matter relative to other genes that were being mutated similarly in prostate cancer? And frankly, we didn't really have models to really make sense of this, and really weren't sure how best to proceed. And so for this study, we kind of left it as is and said, well, we would like to make this molecular signature of metastatic prostate cancer. We didn't really, frankly, solve the problem, but we sort of left it to future work. And that sort of leads into sort of how we ended up going into this direction, which sort of pivots into why did we actually go down this route in the first place? And to do this, what I thought I'd just quickly review is perhaps a bit of an oversimplification of the different kinds of models one can use to try to derive inference from these data and why we sort of went the way we did. And so by this, I specifically mean oftentimes when developing predictive models that were used to make these kinds of inferences, to understand which gene or genes are enriched in a certain context, primary metastatic prostate cancer being the one we care about, there's often a trade-off between models that are very interpretable, meaning you can understand sort of why the algorithm is thinking what it's thinking, for lack of a better way to put it, and accuracy, which is how good is the model at actually discriminating, primary or metastatic disease, for example. And here you have granted, again, a sort of a bit of an oversimplification of the different kinds of models one can use to make these kinds of inference. Everything from logistic regressions all the way down to deep neural networks. And there's a lot of sort of nuance about each of these buckets, and I certainly don't mean to try to summarize that over the course of this presentation, but over the last, let's say 5 to 7 years, there's actually been a lot of exciting advancements in the deep neural network space to try to sort of increase the interpretability of these neural networks that are otherwise often sort of described as black boxes. And over here, I'm listing some of the algorithms that have been developed that try to derive attributions of sort of why did the algorithm make the decision it did, what features did it determine were important in making these inferences, and how could that be used for all sorts of purposes, everything from natural language processing to computer vision and so on and so forth. And so we saw this and thought, we wondered, maybe there was a way to actually combine sort of everything we know about biology into a neural network, and in some sense bake the interpretability into the engineering of the network itself to enable the kinds of interpretation that we'd like to get out of these algorithms, and then apply them to those kinds of contexts like the primary metastatic prostate cancer context I was describing before. And the good news is that we certainly weren't the first to think of this. And actually, there's some really foundational and wonderful work done by colleagues in the field who've applied similar paradigms to different biological questions. And so on the left is an example from Trey Eideker's lab at UC San Diego, deriving sort of what he called a biological neural network to make inference about yeast biology and DNA repair. And on the right is a sort of biological neural network actually for drug discovery, in this case, sort of for looking for different kinds of antibiotics that was developed by the Collins lab at MIT. Again, similarly, in each case, using biology into a network to constrain the network to a point that lets folks understand sort of why it made the decision it did. And so with this as a background, what we then proceeded on to do was build our own neural network for cancer genomic data from patients. And so we called this PNET, and this was developed by Dr. Haitham El-Marakebi, who's an instructor at Dana-Farber and a researcher in my lab, a computer scientist. And the idea here is we would take as inputs any kind of molecular feature that might be relevant, in the cancer patient data that we get. So most commonly somatic mutations and copy number alterations, but it could also be flexible for other kinds of types of molecular events, fusions, methylation changes, gene expression, and so on and so forth. And rather than have a sort of deep, fully connected network that could achieve the best accuracy on its own, but may sort of have struggles with interpretability, we decided to make what we called a biologically informed network architecture, such that each layer of the neural network actually has a sort of a certain kind of a meaning, whether it's genes, pathways, processes, and so on, all the way to outcomes, such that we could train it to ask the question we care about, but then go back and sort of, in some sense, open the box and look at each of the layers and interrogate them to understand why it made the decision it did at each of those layers that we could then use, hopefully, to perform downstream experiments and also perhaps apply to clinical contexts as well in a way that clinicians and others might be more trusting of in terms of why it made the decision it did. In terms of the architecture and then prior knowledge that we decided to encode into the network, we sort of went into publicly available options and found the Reactome database to be quite a useful resource, and that helped us create a series of pathways that with genes, pathways, and processes all connected together that we could engineer directly into this network. Now, we built it in such a way that it incorporated this automatically. One could actually modify this to sort of say, well, I don't agree with this signaling pathway or whatnot, and actually perform your own biological encoding, so to speak, into a network if you'd like. So then what we did, after we did a fair amount of the engineering, is we went back to the same data set that I described earlier, the one we had already published back in 2018, that was split between primary and metastatic prostate cancer, and that had admittedly pretty limited clinical information, and I bookmarked that because I'm going to come back to that in the end. And we asked, well, what if we compared our model to all those other kinds of models that are used for this kind of discrimination or classification task to see if we could do a better job. And because this was a bit of an imbalanced cohort, I'm showing precision recall curves instead of sort of more traditional ROC ones. But needless to say, if you just sort of look at our model compared to others, we did as good or better than everyone else, albeit not by a whole lot. But we felt like this was actually encouraging to suggest that sort of a biologically informed neural network could actually behave and perform just as well, if not better, than any other strategy for this particular classification task. We had taken this data set and split it into training, validation, test sets, as one does in these things. We then went and actually tried to apply it to external data sets that the model has not seen. And we'd say we did a pretty good job here. It wasn't perfect by any means, but when we applied it to independent data sets of localized or metastatic prostate cancer patients, it was actually able to perform reasonably well. And I didn't include this slide in this talk, but I should also add, sort of as a bit of a curiosity, we noticed that actually in some cases where the algorithm had misclassified a primary prostate cancer as metastatic, those patients tended to have far sooner recurrences than the patients with primary prostate cancer who were not misclassified. And I think this also suggests that there may actually be some sort of untapped potential for the use of these kinds of algorithms for prognostics in the clinic, which is something we're actually exploring now. And so all of this was, fun and exciting for us, and sort of demonstrated as a proof of principle that you could take sort of known biology, blend it with sort of advanced principles of computer science, and use it to ask an answer or to ask a clinical question related to prostate cancer genomics, and we can make these predictions. But again, the reason why we wanted to actually pursue this was a little bit less to sort of demonstrate that we can get a model that can maybe squeak out a little bit of performance better than the other models. That was good, but that's not why we really wanted to pursue this. We really were motivated because we wanted to understand which of these genes and pathways were actually sort of doing something to promote metastatic prostate cancer, and therefore, how could we intervene upon it if we can only figure that part out? And so for this, the nice part is again, this is a network that is intended to be fully interpretable, and such that one could inspect every layer of the network to try to understand why it made the decision it did. And so with that, we're actually able to take our model that we had trained on the data set I described and start to inspect each and every layer. And so I'm going to actually sort of show you what I mean. It's sort of a little bit easier than trying to explain it verbally. But basically, here's the first sort of set of inputs, basically mutations, amplifications, and deletions, and how those contribute downstream and converge onto genes. And so here again are some familiar ones. So AR, P53, and P10, just like before, actually still remain in this model to be the genes that are the biggest contributors to making this discrimination between primary and metastatic prostate cancer, assuming they have one of these molecular events in one of these genes. And what was already starting to be interesting was we were noticing some of the other ones actually were a little sort of less on the beaten track, a little unfamiliar to us in context, or were genes that we had heard of, but we weren't sure what they were doing in this particular context. And so then these genes started to converge on certain processes that then actually expanded down into fundamental biological principles. And so in total, let us, in essence, inspect the entire sort of biological processes and the interconnectedness between them that might explain the differences between primary and metastatic prostate cancer. And there's way too much to sort of dive into in terms of how one can interrogate this. Actually, with our study sort of in our GitHub repo, we have an explorable version if you want to play around with it yourself. But what I wanted to call your attention to is actually sort of one of the things we noticed pretty quickly. So again, there's P53, which, you know, not too surprising, sort of a pretty well-known cancer gene, and certainly a well-known prostate cancer gene in metastatic prostate cancer. So when we were looking at this, we said, well, that's great, but what about MDM4? We were not expecting to see this, and in fact, there had been a little bit of work on MDM2 in metastatic prostate cancer, but very little on MDM4, and we then noticed that these were all focal amplifications, and they were all occurring in P53 wild-type settings. And so just to sort of as a very, very quick refresher, you know, what am I talking about here? Well, I'm talking about sort of an unexpected sort of, but known, a sort of a known needle in a new haystack, basically a gene that we know a lot about in a context, in a genetic context that we know a lot about as a field. And this is actually taken from a review from one of Dr. Arnie Levine's reviews on P53 biology, but in a very unexpected space. We really don't think about this in prostate cancer much at all. This was not something that certainly was on our radar, to say the least, and was something that we really sort of could only observe really in the context of the other genetic events that were happening that was informed by the interpretability of this network. And so with that, we then said, well, that's interesting. What can we do with this? And so as it happens, right as we were sort of making this observation, we were also wrapping up and making some contributions to a preclinical study that was being led by Dr. Bill Hahn and colleagues over at Dana-Farber, led by Justin and Rand, pictured below. And what they had done was an ORF screen, looking for genes that, when overexpressed, conferred resistance to enzalutamide in sort of a castrate sort of mimicked cellular environment in prostate cancer cells. And so this is the results of their screen, and we mapped some of the genes that we were observing in our model onto this plot. And among the top hits of their screen was actually MDM4, which was, again, biologically consistent with the genetic profiles we were seeing in our patients. ORFs for MDM4 in this context, focal amplifications in our patients. And so that led us to the hypothesis that perhaps if we could therapeutically inhibit MDM4, specifically in a P53 wild type setting, this might actually have translational relevance for prostate cancer. And indeed, that's actually what we did, and that's what we found, that the P53 wild type cells, the prostate cancer P53 wild type cells were sensitive to MDM4 selective inhibition, and those that were mutant were not. And all of a sudden, we could take an observation from a data set we had already published 3-4 years ago using sort of a more sophisticated analytical strategy paired with some molecular biology as confirmation, and provide justification for, in essence, repurposing a therapeutic strategy that's being tested in other contexts, but not in prostate cancer, and applied into prostate cancer in a genetically stratified way. which we were pretty excited about as a team, and actually is sort of leading into some phase 1 trial designs that we're trying to conceive of now, and sort of hopefully take forward into the clinic in the not too distant future. So where can we go from here? So in some sense, very admittedly, this was a bit of a toy model, and we weren't really expecting this to sort of take us where it ended up. But we already recognized pretty quickly that we're starting with copy number and mutation data, but that's a pretty simplistic view of the molecular complexities of prostate cancer, or really any cancer, any solid tumor. And we'd like to generate more sophisticated, complex, and interdisciplinary data. And so for this, one way we're doing this locally at Dana-Farber is through something called the Center for Cancer Genomics, whereby we're sampling tumors in serial strategies, looking to generate both bulk sequencing data, but also single cell and spatial information from our tumors before, during, and after therapy, and trying to use that as sort of a bit of a foundation to help guide more of these discoveries. But on the algorithms themselves, I think we have a lot of work to do that I think will be a lot of fun to try. And so, for example, we started with mutations and copy number information because that's basically what we had and tends to be the most straightforward kinds of alterations to look at in these contexts. But we know there's a lot more complexity to this. So even within the copy number alteration space, we know that there's value in sort of discriminating between single copy and dual copy loss, or combinations of single copy loss and mutations, and so on and so forth. And in prostate cancer and elsewhere, recurrent fusions play an important role in pathogenesis. So actually, we incorporated these features in supplementary analyses as part of our paper, but we know things get a lot more complicated. And so, for example, and we have a paper coming out later this week, whereby we're looking at both coding and non-coding somatic mutations and what they might mean in prostate cancer or elsewhere, and would like to incorporate these features into such a model. And we'd like to incorporate, transcriptional information or epigenetic information of not just the tumor cells, but perhaps also the immune microenvironmental properties and programs, because we believe those together might also provide a different angle into some of these biologically convergent or divergent processes that help explain different groups. And on the analytical side, one thing we kind of hadn't appreciated was that we were trying to, somewhat unwittingly, solving a bit of an open problem in the cancer genomics world, whereby historically we tend to have different algorithms for significance of mutations, copy number alterations, fusions, and so on. And here we actually provide a mechanism to squish it all together into one thing that we could actually then use for inference. And of course, we just applied this to the setting that we were looking at initially, primary and metastatic prostate cancer. But there's a whole host of questions that we could ask that could really span lots of different clinical contexts, whether it's localized prostate cancer after surgery, which patients are more likely to recur versus others. We have a proposal we've been working on now for quite a while, trying to get funded to try to do that. We'll see how that goes. But you could imagine outside of prostate cancer, or even outside of cancer entirely. We've actually had some collaboration start in the cardiovascular domain, again, using some of these same principles in those contexts. And of course, we'll be very happy to collaborate with anyone and everyone as we go along moving forward. The last part of it, though, is, and I kind of had hinted at it earlier, is that, one sort of very glaring hole in all of this is that our phenotypic information, what we know about the patients we're studying, is pretty limited in these data sets. And historically, that's often because either we don't collect it, or at least in the United States, we tend to capture information from patients from a very few selected hospitals that tend to skew towards certain demographics, socioeconomic status, race, and ancestral backgrounds. And that's not right, and that's not how we're going to move the field forward as a larger community. And so one thing we're trying to do is sort of change the paradigm on how we actually collect these data. And so here is something called the Metastatic Prostate Cancer Project, or MPC Project.org. And the idea is simply put, rather than to have the patient come to us to participate in research, we can bring the research project to patients and to their doorsteps, such that they click the Count Me In button. And then what happens is they fill out an online consent form. We then can send them liquid biopsy kits, saliva kits. We can actually send more than one liquid biopsy kit to actually trace their tumor evolution over time. And use that as a means of data generation that includes patient-reported outcomes, abstracted information from the electronic health record, and sophisticated molecular analyses from a wider swath of patients than we tend to study. And so far, it's pretty cool. We're over 1,100 men who've signed up since we launched a few years ago, even with about a 1.5-year pause in the context of the pandemic, where we intentionally stopped operations so as not to overwhelm the system. We're pretty excited about this because, again, that basically matches that entire cohort I described, which took our team about 8 years to accrue. And even more importantly, to what I mentioned earlier, we think we're capturing a different patient population than we tend to typically study. And so, for example, the vast majority of our patients are not cared for at NCI-designated cancer centers in the United States. We tend to be enriched for patients from sort of medically underserved or physician shortage area populations relative to studies of this kind typically. And we can start to piece some things together that incorporate patient-reported information abstracted from the chart, and genetics, with the hope of then feeding it into these algorithms moving forward. And so here's just an example of self-reported family history relative to chart-reported family history relative to detection of a pathogenic germline mutation in a known prostate cancer gene, actually identified using some deep learning methods. There's a preprint if folks want to check out what I'm referring to in more detail. So with that, right at 25 minutes. Thank you so much for this invitation. There's too many folks to thank from our team and our friends and colleagues near and far. It's a privilege to virtually be here. One day, I hope, get to see you all in person somewhere, and that all are safe and sound in these crazy and unpredictable times. Thanks again for the invitation.
Thank you so much, Dr. Van Allen, for the excellent presentation. So we have a question from the audience. Jeron Fernando was asking, why does the BRCA1 1 and 2 genes are not involved in the AI modeling?
So if I understand the question correctly, like, where were mutations in BRCA1 and 2 in our network? So BRCA1 didn't actually make it very far, but actually interesting, BRCA2. So we could only visualize, for the purposes of what we actually showed in that little figure, we could only go down to maybe like 8 or 9 genes into the list. BRCA2 actually was pretty close to there. It just didn't quite make it under the figure for what it's worth.
Okay, makes sense.
So actually, to your point, it is, as we've known from us and many other people have published on how there is enrichment of particularly BRCA2 in this context, in the metastatic context. And so, we do recover that, but it's not as strong for some reason, at least in this analysis. It didn't make it into that figure, but it's definitely there.
Okay, excellent. So thank you so much again for the excellent presentation. It's an excellent talk. So Dr. Leung, if you want to.
Yes, thank you very much. Really interesting. Just out of interest, Professor Van Allen, the PNet approach, is it possible to apply proteome and metabolomes in that as an extra layer for the analysis, and also within the analysis of primary tumor and metastasis? How do you take into the consideration of subpopulations within each sample?
So great question. So actually on the first question, so in principle, it's flexibly feasible to incorporate any data type you want to include, can be included. It's always with sort of an asterisk and a word of caution, right? So we didn't include gene expression data into this method of this development because we knew there was these like irreconcilable batch effects with the gene expression data that we were dealing with between the primary and metastatic diseases that would result in some catastrophic performance of the model downstream and sort of make it hard to interpret. So we didn't do it there, which doesn't mean you can't do it. It just means you got to be careful, right? And so in principle, those kinds of data would be reasonable. In terms of the other question, so it's a really interesting one. We didn't intentionally include information about clonality, right? Like, is this a clonal mutation or a subclonal mutation to this model? We've since tried to experiment with that a little bit and see if we can make inference. We don't, one of the curious things about these networks is you're sort of seeing what they can learn without you sort of in a, I guess, weakly supervised or almost unsupervised way in some respects. And it seems like it sort of can kind of pick up some of the sort of, perhaps, I mean, this is very speculative, like the clonal pattern, the patterns of clonality and how those things in certain genes might actually matter, right? So I think we didn't include it here as a feature type, but it can be. And I think it may also be learning some of these things, even if we don't directly sort of guide it in that direction, But it's a very interesting area we're curious to explore moving forward.
Thank you.
Oh, sorry, you're on mute.
So yes. So following the previous considerations, so you mentioned in your model that you were considering different alterations. You actually mentioned methylation and amplification, but at the end you just focus on mutations, if I'm not mistaken. Is it because it's more difficult to deal with those kind of alterations, or it's just didn't came out as important as mutations for your model?
So great question. So on our figure 1 of our study, we kind of put a box around mutations and copy number alterations, because in essence, that's what we had. And that's sort of a more ubiquitous set of data we can use for these kinds of questions. As from the previous question, we had gene expression data on, I'd say, like 2/3 of the cohorts that we had mutation data on. But because of some concerns about technical differences between the groups, we were too anxious to include that as part of the model, but it can be included. We just chose not to for sort of our initial pass at trying to do the study. And more generally, the idea is try to incorporate more feature types as we can. So we did include, even though we didn't include the gene expression data, we did include fusions that were detected from RNA-seq, which we didn't think had the same kinds of issues. And there, it didn't end up really contributing a whole lot to model performance or to downstream interpretation, but that was just sort of a specific use case. So we do want to make it more flexible to have more things.
Thank you. So I think we should move on to the excellent talk. So I think we should move on to the next speaker, and it's really a pleasure. I'm really delighted to have Dr. Litjens. He studied biomedical engineering at Eindhoven University of Technology, and then he did his PhD on computerized detection of prostate cancer in multiparametric MRI under the supervision of Professor Joel Barentz at Radboud University Medical Center. He was a postdoc in Germany. He, in fact, got a fellowship from Alexander von Humboldt Society. Then he went back to Radboud University since 2016, and he was awarded as a Young Investigator Award by the Dutch Cancer Society, where he started his own group on computational pathology on prostate cancer. So he, in the beginning of 2022, he published his first publication on expert level AI system for Gleason grading on prostate biopsies, and it was published in Lancet. And this study was done in collaboration with Karolinska Institute and with International Panda Challenge for Gleason grading. And the results were also published in Nature Medicine in January of this year. At the same time, he was awarded with the RC Starting Grant, and with this grant, he's further exploring the use of AI for cancer prognostics behind established human grading systems. And today he will for sure give us more details on this. Thank you so much for being with us today, and the floor is yours.
Thank you very much for that kind introduction. So today I'll talk to you about AI in neuropathology. Subtitle: Do we need the pathologists in the future anymore? And for now, I'll spoil the answer. The answer for now is yes, and I'll also explain during my presentation where that exactly is. But first things first. Well, I'll first start with my disclosures, of course. And I don't think I have to spend a long time explaining this audience the diagnostic trajectory of patients with suspected prostate cancer. But what I want to reiterate is that the analysis of prostate biopsies by pathologists still plays a major role in current day diagnostics and treatment decisions for patients. With a Gleason grading system, I think still widely used across the world. With increasing Gleason grades come increasingly worse prognosis for patients. And for the people who are not aware of this, a single Gleason grade turns into an ISUP grade, or depending whether in the US or not, a grade group. And in the end, that determines generally for a large part the patient treatment. However, and this is a pretty recent study from the Netherlands, 2021, grading is pretty inconsistent. So this is a study among 30,000 patients, prostate cancer patients in the Netherlands. And what they plotted here was the observed proportion of ISUP grade group 1, so low-grade prostate cancer, at various different hospitals. And on the x-axis, the number of prostate cancer cases they handle. And you can see that there are many hospitals which either have a much higher proportion than expected or a much lower proportion than expected, which is probably due to the resident pathologist having a different interpretation of the Gleason grading system. But as a patient, of course, this is pretty concerning that your treatment can depend on what hospital you got your biopsy at. So as a biomedical engineer, what we thought was we can probably do that better if we can potentially enhance the pathology with AI support. And we actually did that in 4 different studies. So the first question we wanted to answer was, can we actually perform automated grading at a decent level? So ideally at expert pathologist level. Then we wanted to see how mature in general is AI for Gleason grading. Is this just a point solution that one group in the world can do, or is this something that you can actually replicate at different sites across the world? And once you have these AI systems, how can we actually help pathologists with these AI systems? And at the end, can we actually do better than our current Gleason grading solutions with AI? And the work I'll be presenting today was mostly done by my 2 PhD students, Wouter Bult and Hans Pinkas, whose work I will be referencing a lot during this presentation. So first off, automated Gleason grading. So how do you do that? This is a study we published in 2020. Most of the work was done in 2018, 2019. And with any AI system, the first thing you need to do is actually try to get data to train your AI system, which was actually quite a big threshold, I think, for many groups in the world to actually build these types of systems. Because you can imagine, here we see a picture of prostate cancer. If I want to have a pathologist annotate all the cancerous cells, and then I do not only need to do that for this small region, but I need to do it for entire biopsies. And then I also need to do it for 5,000 patients, or that ballpark, maybe ideally 50,000 patients or even more. You can imagine that it's very challenging to collect this type of data to train AI systems. So we actually did is we took, I would say, quite a creative approach. We actually started with very coarsely outlined cancer regions, which were indicated by pathologists. So in terms of coarseness, you can see here on the right what that resulted in. And then we trained the first AI model to be able to identify coarsely cancerous regions. But you can already notice that there is a lot of tissue in here that is actually not composing of cancer cells. So that was the first thing we wanted to tackle. And that was something where actually the advantage that I work in a pathology lab comes in. because what we did is we augmented our standard H&E diagnostic stain with immunohistochemistry to highlight all the epithelial cells. And by combining that with this coarse annotation of the tumor regions, we could highlight all the cancerous epithelium. As you can see here, that's something we did in 2019. And once we had all the cancerous epithelium, what we did is we first took all the patients who had pure Gleason grade, so 3+3, 4+4, 5+5, and trained an AI system which was able to predict these different grade groups. And that we used to classify our entire training set of patients, which also included heterogeneous Gleason grades. So that was roughly 6,000 biopsies. And then we used that to completely train our AI system, which resulted in an AI system that does a pretty nice job, at least qualitatively, of highlighting normal epithelium and the different grades of Gleason grades of cancer in biopsies. But of course, a qualitative result doesn't mean that much. So we wanted to look at this quantitatively as well. Here another example of how detailed you can get if you use IHC as a base. So that works pretty nicely. So what we did is we actually asked at USCAP, which is the biggest pathology conference in the world, we actually gave a presentation there and asked pathologists if they wanted to have their performance measured against an AI system. And actually our reference standard here, so the reference Gleason grade, which we compared both the algorithm and the pathologist against, was set by 3 uropathologists in sort of a Delphi scheme, where they first individually graded all the biopsies. So all these pathologists had more than 20 years of experience. Then they were given the chance to correct their results while given also the annotations of the others. And then for the cases where we didn't have a consensus, we held a consensus meeting to make sure that all of them agreed on a single grade for every case. So what did we do in the next step? We compared the AI and the 14 pathologists that wanted to participate against this reference standard. And we first used quadratic Cohen's kappa. And the quadratic Cohen's kappa is an agreement measure where if you score a 0, then you are guessing. So you have essentially chance agreement with the reference standard. And if you score a 1, then you have a perfect agreement with the reference standard. And ideally, you want to be as close to 1 as possible, of course. So what we did was first we looked at the pathologists which had little experience. So a pathologist with less than 15 years of experience in prostate cancer and Gleason grading. And these are the light blue dots. And we also have the group of pathologists who have more than 15 years of experience. And you can already see that at least experience matters. So if you have an experienced pathologist, the chance that they will agree on the Gleason grade is much higher than if you have an inexperienced pathologist. And where does the AI come in? So this was the median pathologist. The AI system did slightly better than the median pathologist, roughly equal to the expert pathologist, so the experienced pathologist, and significantly better than all the inexperienced pathologists. So that was already a nice result for us. Of course, we also looked at meaningful clinical endpoints. So in the paper, we actually looked at different partitions. So we also looked at benign versus cancer. We looked at benign plus grade group 1 versus the rest, and benign versus grade group 1 and 2 versus grade group 3 to 5, which is what I'm showing here. So if we look at our experienced pathologists, these are the dark blue dots again. And then we can also plot our AI system, which actually provides a continuous output in an ROC analysis. And you can see that it's pretty in the middle of the experienced pathologist. However, it could be a bit better. So we would have hoped that it would be a bit more to the upper left, which is a better performance. And the main issue here was that the AI system and the pathologists have a different way of assessing grade proportions, let's say. So the biggest discrepancy was between the 3+4 and 4+3 cases. where generally the pathologists use a different way to measure the volume of these elements than the AI system does. So we could improve that a bit, but overall, we were pretty happy with this because we're still outperforming all the inexperienced pathologists. So that we solve it, well, not exactly. So you can see there's still a lot of discrepancy, at least in the intermediate grades. I think for the grade group 5 and the benign cases, we're doing a pretty good job. But not for everything. So that's why we set out to expand our research a bit. So we actually hooked up with a group from Martin Eklund's group from Karolinska Institute and with Google Health, who were working on a similar topic, and decided to pool all our experience and all our data together to see if we can do better than each of us individually. And better yet, we also wanted to see, because we were cross-continental groups, how well AI developed on one continent generalizes to data from another continent. So do US pathologists grade differently than European pathologists, for example? Are the patient populations significantly different, and does that affect the AI solutions that we develop? So what we did is we pulled together all our data. So in total, we organized a challenge in which you make available all the data that we collected. So 10,000 biopsies from Radboud and Karolinska that were publicly available to all participants, free to download. And their goal was to come up with solutions that were better than our solutions. And to test their algorithm, we had 2,000 biopsies with a consensus reference standard from different sites, so from Radboud, Karolinska, but also from Google Health. And the majority of that was hidden. So the participants couldn't look at the images, they couldn't access the data. They could only, in the end, after all the evaluation, get the results on that data, and we would do the evaluation. And 500 biopsies were visible so that you could actually check whether their algorithms did a decent job on that type of data. So the way we organized it is that there was a development phase. So all the teams had full access to the development data set, and they could optimize their performance over time on that development set. And once that development phase, which lasted for 3 months, was completed, the participants had to submit their algorithm to us, so their AI algorithm. And we selected the top 15 algorithms out of over 1,000 algorithm submissions. And we validated that on 3 extra data sets, which the participants had never seen and were also not able to optimize their algorithms for. So overall, well, like I said, we had 1,000 algorithm submissions. The vast majority of the teams, so every dot here is an algorithm. The vast majority of the teams were getting very high performance, both on the tuning set and on the test data set part that was visible to them. So they were doing a pretty good job in general. And what was surprising to us is that we had worked on this for 2 years, but that participants after 10 days were already getting results which were close to or equivalent to the results we had in our Lancet Oncology paper, and the same held for the Karolinska algorithm that was published in the same issue. And then over the next 80 days, they slightly improved still, so from, let's say, 0.9 to 0.94, which was still pretty significant, but the vast majority after 10 days would already get a pretty good algorithm. So that also shows that the exact AI methodology is not that important anymore once you have good enough high quality data to put into that algorithm. So like I said, what we did was we had to do a cross-continental experiment. So we actually had 5 pathologists from the US and 4 pathologists from Europe. And we actually compared their grading on a set from the EU and a set from the US, 80 biopsies, equally divided among the different grades. And within the European panel, there was a pretty good agreement of 0.85, also in the US, still even a bit higher. But what was interesting is that the individual pathologists from the EU panel also did a pretty good job on the US and vice versa. And if you look at one panel versus the other, you can already see that there was actually a lot of between the different gradings across different continents and across different patient groups. So that was at least encouraging. So that on the other side of the pond, that the way we do Gleason grading is not very different from the way we do it in Europe. And we also looked at how the algorithms behaved. So on top here, you can see the algorithms on the validation set, the test set that they could actually access, that was visible to them. So they did a pretty good job. This is the bar plot, and the individual algorithms are shown on the bottom. And we also evaluated them on this external US data set, so the performance drops a bit, but also on an external EU data set, which is a bit higher than the US. But in general, the algorithms generalize pretty well, especially if you compare it also to the European pathologists. So this was the European set we looked at. So the pathologists on average are doing a worse job than the algorithms. And in the US set, were doing pretty equivalent. So that was encouraging to see. So in terms of generalization, the algorithms are doing as good a job or better than pathologists across continents. So were there differences between the algorithms and the pathologists, which we evaluated? Yes. And this is a plot where we show that every column here is a single prostate biopsy. And every row is an algorithm. So the top part is the algorithms, and the bottom part is the pathologist. What you can already see from this graph is that the reference here is green, and this is grade 1, is that the algorithms were a bit more sensitive. So they picked up more cancers relatively, but were less specific, whereas the pathologists were more specific, but less sensitive. So they actually misclassified quite a couple of grade 1 biopsies as normal. And that for both cases, there were challenging biopsies. For example, this biopsy, which was benign, so also confirmed with immunohistochemistry, didn't contain cancer. Well, quite some pathologists which actually said this was a high grade, so the red box, a high grade tumor. And this was a case which had severe inflammation in the biopsy, so it was a difficult case, but still. But there was also a case here which has grade 1 cancer, which was missed by all the algorithms. So each of the groups had their own challenges. If you plot that in an RSC curve, again, you can also see that. So the algorithms are the blue boxes, the pathologists are the red circles, and they seem to do something complementary. So the algorithms had high sensitivity and the pathologists had high specificity. And I have to say here, the algorithms were not able to tune on this data set. So they could, if by optimizing slightly on this data set, maybe do a slightly better job, but this was what we saw. And I see I have to rush a bit, so I'll go a bit more quicker. So summarizing, we were able to develop our own algorithm, a point solution. We were also able to show that many people in the world could develop AI algorithms that were at the same level of a pathologist and that could generalize across continents and across patient populations. So now you want to put that algorithm into clinical practice. So roughly you can say for AI algorithms, you have 3 choices. Either you use it as a screening tool. So for example, as an independent reader in a screening setting where you have the pathologist and the algorithm operating independently. Or you have the pathologist working together with the algorithm. So the pathologist looking at the algorithm output. Or in a verification setting where the pathologist does their regular diagnostics and then the algorithm only comes in when they think the pathologist made a mistake. And what we think at this point in time makes most sense is to look at an assistive technology. So what we did is we asked a group of pathologists to look at a set of biopsies, unassisted without AI first. And after 3 months, they had to do this again. And this was how the interface looked. So they got the regular biopsy on the left and the AI output on the right. And we also asked them questions about how they liked working with the AI algorithm. And overall, I think many of them were pretty happy with it. And many of them would like to use it and thought it would make them faster and also more accurate. So what is the benefit quantitatively of AI assistance? Well, unassisted, the correspondence to the reference was 0.8 roughly. And AI assisted, they went up the group of pathologists went up to age 7. But most importantly, at least what I found most importantly, is that their standard deviation, as you can see in this box plot, significantly decreases, which means that as a patient, the chance that you get a different result, depending on which pathologist looks at your biopsy, is significantly reduced. Same holds also for volume estimates of cancer. You see that the standard deviation reduces markedly. What we did was, again, we also repeated this after 6 months on a completely different set of biopsies, and we saw exactly the same results. So that was nice to see, so that this also holds over time and on different types of biopsies. So summarizing, where are we now? I'll just rush through this a bit. So we have an algorithm that works. We have an algorithm that helps pathologists when they work with it. So we should look at integration into the market. And as some of you might know, there are actually FDA and CE certified products for this on the market already. But also what we want to look at, okay, can we do a bit more than Gleason grading? So for this integration, and that's also coming back to why we still need pathologists. So all these algorithms are trained on prostatic adenocarcinomas. And I think you probably have 90 to 95% of all prostate cancer cases are that, but not all of them. And algorithms in general cannot deal yet with things that it doesn't know. So to give you a simple example, this is a piece of colon tissue that appeared in the prostate biopsy. And most of the algorithms will signal that this is a prostate cancer because it's not normal tissue, so it doesn't know what to do with it. And obviously, that's not something you want for your patient. If there is an accidental piece of colon tissue that the algorithm says that they have prostate cancer. So what we're looking at in my group is actually having algorithms express their uncertainty on their diagnosis in addition to their prediction, which could help us flag the cases where the algorithm encounters something that it doesn't know. So that's for the integration part. So wrapping up on that, so for the next generation diagnostics, everything we've done up till now has been essentially replicating the Gleason grading system with AI, but it's pretty clear that the Gleason grading system is not the end-all be-all of grading systems. Over time, we have also noticed that there are other morphological patterns that are relevant to prostate cancer prognostics, for example, intraductal carcinomas that are not present in the original Gleason grading system. So what we set out to do was maybe we can predict patient prognosis end-to-end. So without actually first trying to predict the Gleason grade, directly predict prostate cancer recurrence with AI. So what we took was a data set from Johns Hopkins Hospital in Baltimore, which was actually a very interesting data set for us because it was matched recurrence versus non-recurrence on age, race, pathological state, and Gleason grade. So in the recurrence group, you had the same distribution of Gleason grades as in the non-recurrence groups. So if you train an AI algorithm to predict recurrence based on this, you would expect that the algorithm has to pick up things that are not in the Gleason grading system. So what we did was we validated that on a more typical data set from New York University, which had 204 patients and 42 recurrence events. So I'll just skip over this a bit just to save time. But what we set out to do was set an algorithm that had to predict years to recurrence. And what we showed is first in the Johns Hopkins data set, or development data set, that this AI biomarker had significant predictive power in addition to the Gleason grade. And also in a multivariate analysis that this biomarker still had significant predictive power, even when you add things like preoperative PSA, surgical margins, etc. And then in the validation cohort from New York, so completely different set of patients, we validated this again, and this still held true for that set as well. And you can also see that in these survival curves. What you can see, of course, is that after 15 years, it becomes harder to separate these patients. Also because the date from the biopsy to this recurrence here is so long that actually you could question whether this information is present in the biopsy at all. What we also did was in terms of intervisibility, like the previous speaker, we looked at which morphological patterns happened more in cases that recurred quickly versus cases that recurred slowly. And you can also see some things that we already know from histopathological literature, like introductive carcinoma, cribriform growth is associated with worse prognosis, whereas low-grade patterns like small glands with prominent lumina is much less likely to recur. So actually what we did show was we could predict biochemical recurrence end-to-end without an intermediate step of glowing via the Gleason grade. And the signal also stays there in multivariate analysis, where you include other clinical parameters like surgical margins. And that with automated concept explanations, we could actually find interpretable clusters that were associated with worse prognosis. And then I leave you with the thought for next year, maybe, is that now we were looking just at pathology, but of course we have much more data streams coming in for prostate cancer patients, which we in the end could combine to move towards optimal personalized treatment. Now that I'd like to wrap up, so acknowledge my research group, all the people who do all the hard work. And I'd like to thank you all for your interest in being here. And if you're interested in more information, you can look at these different sources. So thank you for your attention.
Thank you so much for this very nice presentation with very interesting and provocative data, I must say. But nevertheless, I think pathologists and AI, they work nicely as a team. And of course, there are some aspects that need to be improved. So we have a lot of questions. So I will start with the questions posed by the delegates. So the first one comes from Lazaru, and he asks, do you believe it would be hard to train an AI system on images during the live procedures? We have an idea in our institute about that. Do you have any experience on that?
Yeah, it depends a bit what you exactly mean. So what we've worked on in my group as well is, for example, frozen sections that are acquired during surgery, but not for prostate cancer, but for example, for basal cell carcinomas of the skin. When you do Mohs surgery, it's a type of surgery where you iteratively remove tissue to prevent scar formation as much as possible. And that works perfectly fine. We've done the same thing also for sentinel lymph node procedures, where they take out a lymph node, you check intraoperatively if a metastasis is present on a frozen section. There you can use AI as well. If you mean like on surgical video, yes, there are groups working on that, but I have very little experience with that.
So, and the following question comes from someone that didn't want to show his or her name. So among all DL models, why did you use UNet architecture?
Yeah, so when we started this project, which was roughly in 2017, that was still mainly considered state-of-the-art for segmentation tasks, so where you had to show the individual cells, let's say. Nowadays, I would use a different model, but in the end, if you look at the results from the Kaggle challenge, the exact model you use doesn't even matter that much. You could make different choices there and then still end up with roughly the same results.
So, and following that question, so how was the AI fed the microscope images? Were the photos made using a software integrated microscope? For sure, it will depend on the software that the images are taken. So that will for sure influence the algorithm. That should be my interpretation from the question.
Yeah, so our hospital has a fully digital pathology department, which means that all our glass slides are scanned with whole slide scanners. So these are machines that take 10,000 photographs of a slide at very high resolution and stitch them together. And that was also the type of data we used to feed the AI algorithm. Is the AI affected by which machine makes these photographs? If the AI algorithm is not properly trained, yes, but there are ways to mitigate that. For example, give it data from multiple devices, strategies like data augmentation for the people who know what I'm talking about. So yes, it could be affected, but you can mitigate that such that AI is not so much affected by the data from the microscope or this scanner.
And following the question that I just read to you, I have a question regards the fact that you are using this mostly for biopsies. For sure, there are many biopsies per case per prostate. So how did you integrate that data?
Yeah, so in our hospital, most of our biopsies are acquired under MR guidance. So we don't have that many biopsies per patient, so typically 2 or 3. In contrast to like systematic biopsy via transrectal ultrasound. So right now, that's not something we specifically integrate. So we just give an AI output per biopsy and expect the pathologist to combine this information into final outcome per case. But that would definitely be interesting to look at if you would give all the biopsies, for example, to an AI algorithm. I could imagine, for example, that if there's one biopsy which has a massive amount of prostate cancer and there are others where there's a doubtful foci, that it could help make those predictions on the other biopsies as well. But it's not something we have tried yet.
So more questions from the delegates. So is kidney biopsy as easy to process as prostatic one?
Well, from an AI perspective, yes, although the clinical questions are completely different, of course, that you would have with a kidney biopsy as with a prostate biopsy. Actually, we have one PhD student in our group working on kidney transplants. So then you look at, is there inflammation, for example, present in the kidney tissue that could signal rejection of the transplant organ, things like that. And that you can also do with AI. But it's a completely different task, so it's hard to say as easy than prostate or not. It's just different.
Yeah, and one aspect that it's also important, since you are analyzing biopsies, you have an area that it's much smaller, for instance, than when you are analyzing fragments from a prostatectomy. So how do you think they might be related? Do you think that you would have more confounding effect due to the fact that you might have more stroma and other aspects related to the fact that you have a full fragment instead of biopsy? Do you have any experience or comment on that?
Yeah, it works both ways. So in prostatectomies, you have pieces of tissue or tissue areas that you don't often see in biopsies. Think of things like the urethra, the stromal connective tissue at top of the prostate. So if you have an algorithm that's trained purely on biopsies, there is a risk that it makes mistakes in these types of tissue. Conversely, if you have an algorithm trained in prostatectomies, in biopsies, you have more tears, more tissue artifacts due to processing of like thin strips of tissue. So ideally, if you want an algorithm that works in both settings, you need training data from both acquisition methods. So you need both biopsies and prostatectomies.
And another aspect where I think it should be interesting, because as far as I understood, all the cases that you have analyzed, all the biopsies were already known to harbor cancer, right? So how does this approach should be working with, for instance, with prostatitis cases or inflammatory conditions, not specifically with glands that harbor cancer?
Yeah, so we had both cases that had proven not cancer and cases that had cancer. In the cases that did not have cancer, we actually in the paper have a table on the benign conditions that were apparent. So we had quite a number of cases which had prostatitis, I don't know if this is the correct English phrase, but granulomatosis inflammation, yes. Or prostatic intraepithelial neoplasia, for example. So we had those cases in this data as well.
Thank you. really nice. And of course, and we have to remind ourselves that pathologists are not only do not only interpret the slides. They interpret the slides based on the clinical data and all the other variables that are relevant, right? But is there a way that you can add that to your algorithm? Maybe it will for sure increase the power of the information, right?
Yes, definitely. So that's also what I wanted to say with my comment on everything we look at right now. And I think that's also a question is prostatic adenocarcinoma. And currently training AI models with the rare prostate cancer types that are occurring like in 0.1% of the patients is still impossible. And we don't have a way yet to have our algorithms deal with these rare occurrences. And that's also why I think pathologists are not going away anytime soon. It's more that if you can get like 50% of the easy cases very quickly processed, they will have more time to handle the more difficult cases. And I know from experience that they get more and more work. The analysis becomes more and more complicated with molecular pathology and different kinds of immunohistochemical staining. They can use all the time they can get.
Yeah, that's a very polite and clever answer. Thank you so much for
You're welcome.
Very, very, very excellent. I must say 2 excellent talks. So the floor is yours, Hing.
Excellent. Thank you very much. I think we've exhausted all the questions, and just want to thank our 2 excellent speakers today and my co-chairs in chairing this meeting, and for all the delegates for joining us this evening. Have a good evening. Bye-bye.
Thank you. Thank you all. Bye.